Thank you so much. And I try to hear that you me. So I'm founder and CEO of Origami Therapeutics, and we started Origami with the idea of functionally curing neurodegenerative diseases. So what I'd like to do first is introduce you to Huntington's disease. Some of you may not have heard of this, but this is a devastating, fatal genetic disease. 1 of 10 most common genetic diseases. Age of onset is as early as average 35 years of age. So think about where you are when you're 35 years old. There you will be or have been. And you're in the prime of life. Your career's going, you've had your kids, you're having your kids. And that's part of why this is so devastating. And the duration of the disease is anywhere from 10 to 25 years. So it's a long haul. Furthermore, this is an autosomal dominant genetic disease. And what that means, if I can get the to work, but what this means is that you only need 1 copy of the mutant gene and you will get the disease. And so what this picture just shows you is that 50% of the children of a couple where one couple has 1 mutant gene, you can expect 50% of those children to be affected. And so on, multi-generationally, in the next generation, 50% of the children of an affected individual. So, symptoms. This disease used to be known as Huntington's chorea based on the movement disorders, which you can see up in the right corner. But there are a lot of other features to this disease that make it particularly devastating. Cognitive decline, mood swings, speech difficulties, hallucinations, behavioral and personality changes, which make it difficult for people to hold jobs and to think about even, how do I get to work? I used to know which bus I had to take and how I get there and whether I turn left or I turn right. And it doesn't happen, and it's hard to follow instructions. So there are no effective treatments. The only labeled treatment for Huntington's disease are 3 medicines, 1 that's generic. They all work by basically the same mechanism, and that is to only partially treat involuntary motor movements. And in fact, I actually know a number of HD individuals who don't even take this because, did I lose it? No, because of the side effects. And so in essence, there's really nothing suitable for these patients. The other thing is we think about this as a brain disease, neurodegeneration, and it certainly is because the brain is the most sensitive to this mutation. However, we feel that you require whole body treatment because there are many different organ systems, immune system, GI tract, reproduction, glucose tolerance, so your pancreas, many different, and muscle atrophy, as you can see in the picture on the right. And so there really is a huge need to treat not just the brain, but the rest of the body as well. So, what does the disease look like, and who do we think we might be able to treat? And I get this question often because sometimes people think about the folks they know who have dementia, and when can you even treat? When's too far? When is too late? So, because it's a genetic disease, we actually have the unique opportunity here to actually get in early if we have something that has sufficient benefit to risk ratio, and that's what we're aiming for. And so the prodromal folks don't yet have symptoms, but we know they are gene positive. So we have the chance to both prevent, halt, and slow the disease. Once symptoms appear in the early and middle areas of the disease progression, we have the ability to halt and slow disease. And that actually is really the value proposition that we're after here. So, what is this mutation? So this is a mutation in a gene called Huntingtin, aptly named, And what you see, what is being shown here on the top is a normal gene, and it has under something called CAG repeats, which appear in your genome. And so there, the neurons are healthy, and with aging and stress, they're able to maintain cell homeostasis and health. However, if you have a mutated Huntington protein, as shown on the bottom, you have a longer repeat that affects the protein that's made of Huntington. You have an impaired neuron, it functions for quite a bit of time, which is why this is a later onset disease, not a childhood disease necessarily. But neurons just aren't resilient enough to be able to handle aging and stress. And so an example here is shown on the right there of the HD brain, where you're really missing quite a lot of the brain volume. So how do we solve this problem? The goal here is to selectively destroy the mutant Huntington while sparing the normal protein. And that's critically important because the normal protein is critical to neuroprotection and to normal function throughout the body. So every single cell in your body, from conception to death, expresses this protein. It's critically important. So you just can't live without it. And so our goal is to selectively destroy the mutant protein. So let me tell you a little bit about how our compounds work. On the left here is the normal protein, and in biology, shape equals function. Sometimes you hear that for furniture too, but in this case, shape equals function. And so if you don't have the right shape like the normal protein does, you don't work as effectively. So the disease protein shown just to the right of it, it can properly fold and it can properly work. However, as you age, you're less able to keep everything properly folded and everything working well. And so the protein comes unfolded and can't fold back. And that's shown in the middle there. And so what we have found is a small molecule that actually binds to that misfolded protein and can basically take it out. So it basically binds to the protein, the mutant Huntington, and takes it into the natural cell degradation pathway. So that's basically how it's working. So one of the things that we do at Origami is to make sure that we're using patient-derived human cells. And so a lot of times you'll hear about therapeutics being tested in mice, and then it doesn't translate to the clinic. What we found through my experience and others is that testing in human cells is critically important. And we've been able to show both that our molecule, shown in the graph here, is able to reduce the mutant Huntington and not touch the wild type. More vividly exemplified on the right is the fact that the disease neurons, shown in the middle, clump together, and that's where you're seeing the arrow and the sort of blue balls. Those are the nuclei, and that's distinctly different from the left panel, which is the normal. When we treat the disease neurons with one of our compounds, we can actually prevent that malformation, and in a number of other factors that we've looked at, prevent and reverse them. In addition, we can show that this compound is able to get into the brain and do what we needed to do in the brain, namely lower the amount of mutant Huntington protein, and that's shown in two different brain regions here, the striatum and cortex. Those are the two brain regions that are most highly affected in Huntington's disease. So, who are we? So, beyond being founder and CEO, I have 25 years of experience in drug discovery, primarily in CNS. I put greater than 30 clinical assets into the clinic and 4 marketed drugs for cystic fibrosis. And this approach has been highly based on both my CNS experience and the important lessons that we learned in bringing forward these cystic fibrosis drugs, which have absolutely transformed the way the disease is treated and the prospects for patients. And that's because we went after the root cause of the disease, the underlying cause of the disease, and fixed it. I'm joined by my co-founder, Leslie Schultz, who has a lot of experience in finance, a great chemist that's been with me through the whole process, and some support from BD. We have great KOLs, key opinion leaders. Judy Corey Bloom actually heads up the Center of Excellence for HD Clinic here at UCSD. Steve Finkbeiner, who's up in San Francisco, and a former colleague of mine, Kalpana Merchant, who's very well known in the neurodegeneration field. So I had to have a slide with some words on it. That's to get across the point that this is an orphan disease, subject to expedited regulatory path. There are about 185,000 patients worldwide, but even just in the US, where there are 45,000 symptomatic patients, there are 200 to 250 individuals who are at risk based on their family history. And so it's likely that about 50% of those individuals will eventually develop Huntington's disease. So just in 2020, $1.3 billion for these movement symptom drugs that I mentioned earlier, and they don't even work very well. And so we've had folks estimate for us, but also there are reports out there suggesting that the projected global HD market is over $9 billion worldwide by 2030. So let me tell you a bit about our competition. So this looks like a complicated slide, but I'm going to walk you through this same slide and narrate for you where we are. So one of the things about the brain is, if you have something that doesn't get delivered very well, or you have to poke holes in the brain in order to get your drug in, this is a delivery issue. And it's a problem because not everybody is near a neurosurgical suite and a neurosurgeon. So the access or the democratization of the access is very limited. The other piece is that those approaches have limitations in getting into all the cells. And remember, I told you we have to treat all the cells. So if I take everything away that requires invasive delivery, here's what we're left with. And then if I further take away those approaches that are not mutant huntingtin selective, remember I told you, you have to maintain normal levels of activity for the wild type normal. And so here's what we're left with. And furthermore, Arvinus and Anima actually don't get rid of the most toxic species of this protein, but Origami does. And so we think we are very well positioned to have preferred characteristics for our molecule. So let me tell you about our past funding, about $4.8 million. Some of that has come from phase 1 SBIR, as well as Kickstarter grants from a chemistry outfit, O2H. And we were a Y Combinator batch of winter 22. And the remainder of the money comes from some other YC founders, high net worth individuals, and founder and co-founder. So what we're asking for is $600,000 to round out our $2.5 million seed. We've already closed $1.9 million of that. And what we're aiming to do here is to get to show disease modification. And that will tell us that what our compounds are doing really have a good chance of being effective. And the orange arrow just points to where we are right now and gives you an idea of where we're headed from here, with the idea that in about 3.5 years, we would have clinical data in patients, understanding the dose and seeing some initial proof of concept that our compounds work in patients. So how will we exit? This is a little bit different than what you've heard so far in terms of exits. We don't necessarily have to launch or market in order to get an exit. It's likely either by acquisition or out licensing, and that could come anywhere at development candidate nomination, IND approval, the start of first in man, or the end of phase 1. And obviously, the closer you get to having clinical data and positive data in patients, the more value you will derive. So we're looking at something between 2x and 5x or more return on investment. So just in summary, small molecules, degraders, targeting mutant Huntington, sparing normal Huntington, orally available, brain penetrant, and has favorable drug-like properties, and we're asking for $600,000. Thanks so much.
Thank you so much, Beth. We'll take some questions from the audience.
I think it's great. I just am curious, do you think Origami will work in other expansion repeat disorders, like some with the CGA repeats or even maybe other repeats?
Yeah, great question. Thank you very much. We are looking at that. So what we actually, so everything is Origami owned. We haven't licensed it from anybody. We started from scratch. So we have a number of different chemical series, and some of them work on a similar mechanism to our lead. And so we're actually actively looking at that. So it's possible. And not just CAG repeat, but it may work for other proteins that cause neurodegenerative diseases.
Okay, thank you, Beth.
Thanks.